Hey class, I wanted to do a little clip on both the process of meiosis and fertilization combined so you can see how inheritance works. So first of all, I've drawn already the meiosis for both parents for the formation of gametes from one parent, the sperms on the left and the eggs on the right. Now notice that this is the meiosis of germline cells, and the first thing that happens here is they're going to undergo their own replication of DNA. So this is their own S phase before they go through meiosis. Now remember, they don't go through mitosis, they go through meiosis. So at the end of that S phase, you've got replicated pairs of homologous chromosomes. So you've got two pairs of sister chromatids. Okay, now they both had originally a pair of homologous chromosomes, represented by the capital A and the lowercase a, which means that the genotype of these parents is heterozygous because they have both a dominant and a recessive version of whatever gene A happens to be, whatever trait it happens to code for, right? Now, I did also draw them in different colors to imply that they got one from their mother and one from their father, okay? So you can actually track a lot of where these genes are coming from, where these alleles are coming from, and what type of allele these are if you just color code the allele in the germline cell and as well as in the product of meiosis, so the gametes at the bottom. Okay, so you can see that after the first meiotic division, so here you end up with, you separate the pairs of homologs, so you end up with one pair of sister chromatids on the left and another pair of sister chromatids on the right, and same with the egg. Now remember that the egg, only one of the egg products is going to be viable, so the other ones actually are polar bodies, are not going to produce viable eggs. Only one egg is produced per meiotic event in the oogenesis. Now, after the second division in meiosis, there is no further replication of DNA, so it's just a separation of the sister chromatids. Now, for the sperm on the left, you can appreciate that each gamete is actually going to have one of the alleles from the replication event. Okay, now two of these are the same. Okay, so two alleles are the same to each other, because in the replication of sister chromatids you get just really the same allele for that gene. I didn't draw the chromosome. Okay, the other thing I haven't drawn in this clip is the crossing over event. I just wanted to keep it simple. Okay, now what does fertilization mean? Fertilization means to take any one of these gametes and combine it with the gamete from the other parent. Okay, so let's just randomly select one of the gametes from the father. So one of these sperms over here. Okay, so we take one gamete. Okay, and then we're going to take the only viable gamete from mom, and we're going to do the, what are the possible combinations that you can get from this particular gamete, right? So the fertilization product of combining these gametes, okay, are going to be uppercase A that you get from mom and lowercase a that you get from dad. Okay, now it also, because I'm color coding, I'm also showing that both of these alleles are actually the maternal alleles from both the father and the mother. So these are alleles from both grandmas. All right, so this is where they're coming from. So you can actually get a lot of information from the generations if you know the genotype of the offspring. Okay, now this is just one possibility. So what a Punnett square does is it actually helps you create all of the possible combinations if you were to choose any gamete, and of course also if you were to get any gamete from the mother, because right now I'm only showing you one egg, but you could get 4 different eggs, right, depending on which direction I drew the arrows. Okay, so the mother can generate either this type of gamete, okay, or the mother can generate also this type of gamete. Okay, so that's the alleles that the mother has, and so if you're going to replicate these alleles and separate them into gametes, you're going to get either a gamete that contains the uppercase A, the dominant version of this gene, or a gamete that contains the recessive version of the gene, the lowercase a. Now, for the father, the same thing, except the situation is inverted. For the father, the uppercase A is going to be coming from his dad, and then the recessive allele is going to be coming from his mom. Okay, so just so I can just do this all in once, since I have the color here. Okay, and here. Okay, and let's do the other color to keep it together here. And okay. And the other A is coming from mom. All right. So if I were to use all the possible gametes that can be produced by each parent, I will get the following Punnett square result. Okay. So these are the ones on the top that are coming from mom. Okay. And you can see those are the possible gametes that we produce from the meiosis of that particular oogenesis, and these are the possible gametes that we'll get from dad from the meiosis of dad's germline cell for the sperm, for the spermatogenesis from that. Okay, so those are the possible gametes. This is what these are. These are possible gametes. Okay, and I'm writing the allele for those gametes because right now I'm just sort of concerned about this particular gene, whatever the gene A. And these are possible gametes from mom. And what you get from the Punnett square are the possible combinations. Okay, so I'm going to read them off to you. One of these zygotes is going to be your homozygous dominant. One of these zygotes down here is going to be your homozygous recessive, and two of these zygotes are going to be heterozygotes. They're going to have one dominant and one recessive allele. Okay, now that's the genotype of these. Okay, however, the phenotype. Okay, so I'm going to use black over here. The phenotype is different. The physical manifestation, if this was complete dominance, will say that all of these three out of the four offspring, so 75% of the offspring are going to look like whatever the dominant version of this allele will be, right? So it's going to be dominant phenotype, and only a quarter, so 25%, is going to look like the recessive version for this particular gene. Okay, so this is both an example where you can see both meiosis together with inheritance and the possible gene expression for this allele all in one example. All right, so let me know if this was clear. If you have any questions, I'll be happy to do this again. This is the simplest case, the simplest version for this combined meiosis and inheritance example. I didn't do any crossing over, and I only focused on one single gene, so assuming this is one single chromosome. Okay? Do let me know whether this made sense. Thank you.